Science supports healthier, happier, and harmonious life.
Good morning, everyone. My name is Jun Wang. I'm a principal investigator at the Institute of Microbiology, China Academy of Science. Today, I'm extremely honored to present one of our recent previews or perspective that I have written for the Journal of H Life. First, I would like to introduce you to the Journal H Life. H means healthier, happier, and harmonious life. The journal aims at a multidisciplinary research of biology and medicine, especially the new directions of the challenge we face in the new century, especially with regard to microbiology, medicine, as well as One Health. We sincerely invite you to submit your cutting-edge paper to this journal. Also, we welcome previews, reviews, and also perspectives. Very recently, been privileged to be invited for perspective for H Life. It became online just a few days ago. If you're interested, you can go on the website and check the paper. I'm going to give a short introduction of what this paper is about, and we also hope that some of you might be interested in discussion or even collaboration with us on this roadmap. Protein space is actually extremely vast and it's beyond our imagination. So since at each position of an amino acid there will be 20 at least possibilities, the number of possible proteins of 6 or 7 amino acids would already be over 2 billion, and that's already much bigger than the currently known number of proteins in the database, such as RefSeq. So, what's happening with the rest of the protein space? As a matter of fact, we believe because of evolutionary history as well as some constraints on the protein structure as well as function, the vast space of the protein is not explored by organism, by life yet. So in the evolutionary history, what happens is you have diverse protein families that come from limited number of rows, and they just follow a evolutionary path to a better and better optimum of fitness along the evolutionary path. But it ignores or it doesn't have the chance to explore other species in the protein space that might have even better fitness or totally different protein structure. So I have this metaphor of the protein space being a giant desert. I have this metaphor of a giant desert with only a few plants here and there. Those live plants or those alive plants represent the current protein sequences, and their roots represent the ancient proteins where they originated from. As you can imagine, the number of possible plants that can grow in the deserts are much larger than the ones we have. And the interesting question comes now: would those protein sequences that we have never encountered before have a particular function that we can use for biology and medicine? The answer is yes, because there have been already separate endeavors to explore unnatural or totally random proteins. I have mentioned a few published studies in the perspective that have either used directed evolution, that is, you mutate a few sites on the protein backbone and gain new proteins with even better fitness or novel functions, or there have been studies that express totally random peptides in E. coli, and they observed a decrease or increase in the fitness of the bacteria. Thus, it's very likely that among the species that have not been explored, there are a large number of proteins have functions similar or even better to the currently extant proteins. How do we explore such species? Because the space is vast, and as many scientists, including David Baker, have pointed out, it's nearly impossible to exhaust all the protein species to study each of the protein's functions. However, There have been several advances recently in the field of artificial intelligence, including natural language processing, and we have been exploring the new approaches to identify or understand the functions of new proteins. Recently, we have published one paper in Nature Biotechnology that we used natural language processing to mine antimicrobial peptides from a vast number of metagenomic sequences. This represents one of the endeavors that match the sequence to the function of a protein. And in a few weeks, there will be another publication from our group, in which we studied or which we mined anti-cancer peptides with a similar approach. So it's possible to link the protein sequences to their functions via an artificial intelligence approach. And once we have a better and better model that includes or covers more and more protein families, it's possible that we can follow a roadmap that has been laid out in this perspective and explore step by step the untouched protein spaces. I'm going to give some details on this roadmap. It consists of 3 parts. The first step is actually to explore proteins that have been already sequenced from genome, but with unknown functions yet. The biggest pool of such proteins actually comes from metagenomic sequences, including microbiome, virome, and also fungome or microbiome populations. In this kind of data sets, we encounter large number of small proteins, and sometimes they are termed dark matter of the microbiome. They are very small in sequence length, and many of them, or most of them, actually have no known function yet. But with natural language processing and specific models, we can identify a large number of functional peptides, such as antimicrobial peptides, anti-cancer peptides, and even peptides that are capable of modulating host immunity. This would be the first step that we go to sequenced but proteins with unknown functions. The next would be we change the proteins that have already been known and use a similar approach to direct evolution, and we change the amino acids on a backbone of already known function protein. For instance, we can start from one peptide with already known property of antimicrobial, and by introducing random mutations on specific sites, we can explore the protein spaces that are near to this extant protein. In this process, we can measure the effects of mutations on the peptide itself, especially the property or the potency of its bactericidal or bacterial inhibitory effects. By collecting large amount of such data, we can construct a fitness landscape such as this one, and each pink spot means a new protein that has been mutated from original protein that is represented here with a green dot. Gradually, by establishing a model for each round of data, as well as introducing the next round of mutations, we are very likely approaching a global optimum that is a very potent protein that is from originally a protein with function but relatively low fitness. Okay, so the last step of the roadmap is actually generating and testing totally random proteins. Totally random is very hard to define because once you have a protein sequence and you compare to the database, you would certainly find some extent of similarity to a known protein. So the definition here is the way it is produced. We do not rely on any known protein for backbone or starting point, but we just throw in some random seeds. Once we covered enough possibility or mutation, it's likely we cover a certain proportion of this known protein landscape. And by this kind of sampling, we can step by step figuring out the landscape of this protein fitness. And from this fitness, if we have a very good formula or model, we can directly go to the global optimum and skipping the middle part of directed evolution. You may ask, what is the purpose or what is the utility of such a roadmap? Why should we go to the places that evolution has not touched? Well, I'm just going to give one example of antimicrobial resistance that is increasing very fast in a number of bacteria. The World Health Organization has defined a list of bacteria that is a great concern to global health, as they have the most severe antibiotic resistance, and the rate of resistance is still growing. So by 2050, it is estimated that the biggest cause of global death would be infection from antibiotic-resistant bacteria. The development of resistance toward antibiotics is growing much faster than our speed of developing new antibiotics. So we must think out of the box and go to places that evolution has not touched, and go ahead of the evolution of resistance and develop new antibiotics in a much faster speed. If we have a very large protein space that if we have a method that can explore the protein space relatively efficiently, and we have a good model that can discover new antibiotics in a relatively good speed, it is more likely that we can generate new antibiotics, including antimicrobial peptides and other molecules for treating infections from such organisms. Dealing with such threats from bacteria, as well as other metabolic disorders, cancer, as well as cardiovascular disease, central neural dysfunctions, would require such advances in methodology as well as research paradigm. And I believe exploring protein spaces would be a very useful method for developing new drugs for the future use and treatment of such diseases. About artificial intelligence and more specifically deep learning or machine learning, we are actually at the third wave of so-called artificial intelligence development. The first two waves were respectively in the 60s and in the 80s, but both failed because there were limits on the software, there were limits on the hardware, and more importantly, there were too many promises and non-delivery. But I think the third wave might be different, especially in the field of medicine. Development of neural networks as well as other methods have already gave very exciting examples of how much they can do and achieve in the field of medicine. First, in the section of medical imaging, the diagnostic accuracy of machine learning approaches has already surpassed the accuracy of senior experts in medical imaging, such as images for the eye and images from CT scans. And for dealing with medical records, natural language processing is also very powerful in summarizing and predicting the risks of patients towards several different diseases. So I believe that machine learning, or in general the artificial intelligence, would have a very big impact or even paradigm changing effects on the study of biology and also development of medicine.